Good morning. My name is Prajwal Podel, Product Development Scientist at ACROBiosystem. Before I begin, I want to thank the event organizers for hosting this wonderful conference. And today I will present ACRO's effort in developing critical needs reagents and assay kits to fight COVID-19 and SARS-CoV-2 variants. The COVID-19 pandemic has infected 112 million people worldwide, with 2.5 million deaths to date. The viral agent of this pandemic, the novel coronavirus SARS-CoV-2, belongs to the beta coronavirus family, which includes the SARS-CoV from 2002 outbreak as well. The SARS-CoV-2 viral particle consists of single-strand positive sense mRNA wrapped around the nucleocapsid or the N protein. The genetic material is enclosed by a lipid membrane coated with these membrane protein and envelope proteins. And the defining feature of SARS-CoV-2, as with any other coronavirus, is these projections of spike glycoprotein trimers. SARS-CoV-2 enters the host cell via its spike protein binding to the human ACE2 receptors. The internalization is the key step allowing the virus to enter host cell cytosol, hijack the cellular machinery to translate, transcribe, and replicate this genome. Translated viral structure proteins are packaged in the ER, assembled, and released from the host cell. Now, the current vaccination strategies of Pfizer, BioNTech, Moderna, and the recently approved Johnson & Johnson are all centered on blocking this S protein ACE2 interactions via immunization with S protein mRNA or adenovirus vectors in the case of Johnson & Johnson. While Eli Lilly and Regeneron's monoclonal antibody therapy bind to the S protein to block the ACE2 binding as well. Other human receptors have been identified to bind S protein, including ASGR1, Cremin1, Neuropilin-1, and AXL, which need further research to understand the significance and the mechanisms of these receptors. Antivirals are also being investigated to inhibit specific steps in the viral entry, replication, translation, and assembly. And so far, remdesivir has received a FDA EUA which inhibits viral RNA polymerase for COVID-19. ACRO has developed a variety of high-quality COVID proteins to facilitate the diagnostics and the therapeutic developments against COVID. Our catalog contains three main SARS-CoV-2 structural protein: the S protein, the envelope E protein, and the nucleocapsid N protein. The S protein is primarily expressed in the HEC293 cells. It includes His tag and biotinylated AVI tag. The N protein is expressed in both E. coli and HEC293 cells. N protein is heavily phosphorylated, leading to structural changes that increase affinity for RNA. Due to high number of N protein present during infection, antigen-based diagnostics based on the N protein have been mobilized during this pandemic. E protein is the smallest structural protein of SARS-CoV-2, and we express this in E. coli and with a His tag and GST tags. In addition to the structural protein, ACRO also carries a number of non-structural protein of SARS-CoV-2 that are all expressed in E. coli with His tags for therapeutic development. The SARS-CoV-2 spike glycoprotein is a fusion protein trimer comprised of two subunits. The S1 subunit contains the N-terminal domain and the receptor binding domain to which binds to the ACE2. The S1 subunit is a dynamic structure with closed and open conformation. The open conformation will bind to the ACE2 receptor. Here is the closed conformation shown. The S2 subunit comprises of the fusion peptide repeats, cytoplasmic domain, as well as the transmembrane region. In the trimeric form, the S1 forms the head, supported by central stalk comprised of the S2 subunit. And the high transmissibility of SARS-CoV-2 compared to SARS-CoV has been posited to occur due to the insertion of a polybasic cleavage site between the S1 and the S2 subunit. This reduces the stability of the S protein and facilitates conformational change required to bind to the ACE2 receptors and viral membrane fusion. Prior SARS-CoV research had already established the S protein to be highly immunogenic and key to the viral infection and propagation. And ACRO provides the S protein derivatives, including the S1 subunit, S2 subunit, as well as the NTD, the CTD, and the RBD domains, with the AVI and his biotinylated AVI and his tags and FC tags for wide range of detection and diagnostic assays. ACE2 binding to S trimer initiates the SARS-CoV-2 viral entry. So looking at the structure or docking the ACE2 to the S trimer, it shows that ACE2 binds to one S trimer. Now, ACE2 has also been shown to dimerize, mediated primarily by the collectorin-like neck domain. And ACE2 binds to the open conformation of the receptor binding domain in the S protein with the physical contact on the receptor binding motif. It remains to be seen whether clustering of the S trimer and the ACE2 dimers occurs and enables fusion of the viral membrane to the host membrane and facilitates the viral entry. ACROBiosystem offers human as well as mouse, cynomolgus, rat, and civet ACE2 with a variety of tags, including HIS, AVI, and FC tag, for continual study of binding and assessing efficacy of vaccines and therapeutics. Prefusion stable S protein are needed partly to increase the recombinant protein expression with homogeneous preparations, but also to improve the vaccine efficacy and therapeutic antibody screening. 6-proline substitution has been identified to stabilize loops and cap helices in the prefusion state. And ACROBiosystem offers a high-quality, superstable S trimer, improving upon the 2-proline mutated trimer. This 6-proline mutated protein also contains a T4 fibritine trimerization motif to induce the S protein trimerization, a double alanine mutation to abolish the furin cleavage site, and the S trimer has shown high stability and high homogeneity as seen in this negative stained electron microscopy and Moll's analysis. Functional binding with human ACE2 shows comparable binding affinity to literature values. In addition, comparing the S trimer performance in antibody titer measurement showed ACRO's superstable trimer having the highest signal-to-noise ratio compared to other available S trimers, including the true proline mutant and a competitor S trimer. Such high performance will greatly improve vaccine immunogenicity evaluations. The SARS-CoV-2 infection elicits an initial innate immune response to combat this increasing viral load, which allows the adaptive immune system to kick in and drive down the viral load and clear it out of the body. In mild to moderate cases, the viral load is leveled by the innate immunity and then driven down by the adaptive immune response. In the adaptive immune response, you have the T cells and the antibody response. If we look at the categories of antibody response, it's initially dominated by the IgA antibody, followed by IgG and IgM response. The IgG response persists several weeks to months after infection, thus protecting against repeat infections and also enabling serological tests to detect past infections. ACRO's development of reagents and kits has had measurable effect in supporting rapid vaccine and therapeutics development from discovery to preclinical to clinical therapeutics. In fact, we have helped NIH researchers identify novel protective nanobodies that block the ACE2S protein binding with potency as high as 1.11 nanomolar. Additionally, antibody-like monobodies discovered using ACRO's catalog of S protein and ACE2 were found to neutralize SARS-CoV-2 infections in viral E6 cells with the IC50 of 0.5 nanomolar. Additionally, ACRO's S protein RBD were utilized in assessing immunogenicity of Pfizer-BioNTech's RBD-based mRNA vaccine candidate in a phase 1/2 clinical study. In the diagnostic space, ACRO's support in development constitutes of antigen and antibody detection using lateral flow assay and ELISA methods, which are sensitive enough for asymptomatic and symptomatic patient samples, and also for protection confirmation assays. We support immune response characterization with our antibody detection and antigen testing using our catalog of anti-N and anti-S protein. S1 protein were used to assess IgG subtype titers in hospitalized pediatric and adult patients in Sweden. Lastly, ACRO also supports population screening to assess viral spread, exposure, and vaccine efficacy. ACRO's N protein was used to determine distribution of SARS-CoV-2 exposure in healthcare workers and demonstrated that a significant risk is associated with direct patient contact for these healthcare workers. ACRO has developed antibody match pair for lateral flow assay against the nucleocapsid and the spike proteins. These antibody pairs have high stability, wide dynamic range and assay sensitivity, high reproducibility, and are specifically designed for serological test development. The nucleocapsid detection pair LFA assay shown here performed extremely well with the positive test line at tissue culture infectious dose value of 10. You can see a line here, although it's a little difficult, but nonetheless highly sensitive. ACRO also has a list of antibody available for SARS-CoV-2 antigen testing. Here we have anti-S1 antibody with exquisite specificity to the SARS-CoV-2 when compared to the spike protein from other coronaviruses. Similarly, and the anti-nucleocapsid protein antibody also showed exclusive specificity to SARS-CoV-2 nucleoprotein. And ACRO offers a variety of different type of antibody, mouse antibody, human antibody for both the S protein and N protein to be used in double antibody sandwich assays. In addition to antigen testing, ACRO provides catalog of S protein RBD, S trimer, S1, E protein, N protein for detection of total and IgG, IgM antibody via sandwich ELISA methods. We also provide control antibodies, including NTD and RBD neutralizing antibodies to act as benchmark for neutralization antibody tests. ACRO's catalog of ELISA serologic kits for antibody titer detection offers a wide range of targets from N protein to S protein trimer and S protein RBD. ACRO's kits enable a fast and easy viral antigen detection with high signal to noise and accurate quantification. Additionally, we also provide detection kit for total anti-RBD antibody titer detection anti-S trimer IgG titer detection, anti-RBD IgG1, 2, 3, and 4 detection, as well as anti-IgM titer detection for RBD and S1 proteins. Our TAS-K022 Neutralization Kit is based on a competitive ELISA principle. The neutralization assays approximates the gold standard print assay, which uses live viruses and cells requiring a P3 level laboratory and is a complex low throughput assay. Using a neutralization kit offers an accessible and high throughput method to detect presence of protective antibodies. Therefore, we have mobilized ACE2 incubated with ACE2 plates that can be incubated with the serum samples, followed by HRB tagged S protein RBD. The signal intensity of the ELISA is inversely proportional to the presence of neutralizing antibody. This kit provides qualitative indications and semi-quantitative information and shows a good dynamic range and high sensitivity with the control antibody sensitivity at around 10 nanogram per mil, and also a high lot-to-lot consistency. We use the kit to test against vaccinated serum samples for the presence of neutralizing antibody using 10 vaccinated serum and 70 non-vaccinated serums. And the kit performs spectacularly with a specificity at 98.5% and 100% sensitivity. In addition, our neutralizing kit compared with the FDA EUA approved neutralizing kit showed a strong correlation as well. Now, in addition to ELISA kits, ACROBiosystem is proud to offer the first-in-class S trimer, S1, and RBD protein pre-coupled magnetic beads for antibody screening in therapeutic applications. The magnetic beads reduce time and cost, as well as are convenient products for research and development. ACRO has done extensive stability testing to show low non-specific binding and consistent performance across multiple freeze-thaw cycles. Due to high transmissibility of SARS-CoV-2, multiple variants have emerged around the world. The variants with mutations in the spike protein particularly pose threat to existing vaccines and therapeutics. One of the variants, the UK variant, is now dominant strain in the UK, Oceania, and climbing in Europe. It has multiple detection in the multiple deletion in the N-terminal domain and one mutation in the receptor binding domain of the S protein. It also has several mutations outside of that in the S1 subunit and the S2 subunit. Similarly, the South African variant is now the dominant strain in Africa. It has 3 mutations in the RBD, deletions similar to the UK variant in the NTD, and particular concerning mutation is the E484K mutation, which has been identified to cause vaccine resistance. The Brazilian variant P.1 is a branch of the B1128 variant, which also contains similar mutations in the NTD and the RBD and receptor binding motifs. This variant is attributed to SARS in cases due to reinfection in Brazil. So therefore, these emerging variants have a profound effect on vaccination campaigns currently. For example, looking at both the UK and the South African variant, it is clear that for both Moderna vaccinated serum and Pfizer vaccinated serum, the IC50 fold change from a neutralizing assay shows a drastic resistance or in the potency of the vaccinated sera against the South African mutant. This is again thought to be caused primarily by the E484K mutation, which shows most serum that are less than or greater than twofold resisting to the mutations to the vaccinated sera. Leading vaccines, other leading vaccines with strong efficacy against the original D614G strain have shown lowered efficacy for emerging variants, particularly for the South African variants that you can see here. So the Oxford AstraZeneca vaccine famously was recalled from South Africa due to little to no protection for the South African mutant. Other vaccines show partial efficacy, like the Johnson & Johnson vaccine, with a protective efficacy of 57%, down from around 72% for the D614G strain. So when you look at the neutralizing antibodies, including the therapeutic antibodies that are currently approved from Eli Lilly and Regeneron, shows similar case of resistance from the South African mutant. This has been caused once again by the E484K mutation, and surprisingly, most of the NTD-directed neutralizing antibody show total abrogation of activity, indicating that these mutations and deletions in the N-terminal domain also impact the activity of these neutralizing antibodies as well. Therefore, ACRO has developed more than 60 mutant products covering most of the emerging variants of the UK, the South African, and the Brazilian variants. ACRO is developing, in addition to the wild type, ACRO is developing the NTD single mutants, as well as RBD single mutants associated with these variants, and also the S trimer that contains all the mutation for the three leading variants. These mutation catalog of mutant proteins are aimed to bolster research in the vaccine and therapeutic development against COVID-19. ACROBiosystem has a global presence with the offices in Beijing, Shanghai, West Coast and East Coast of the United States. We have more than 5,000 customer base in over 60 countries, spanning from pharma, biotech, CRO, diagnostics to academia space. ACROBiosystem currently supplies recombinant proteins, including mutants, antibodies, kits, and pre-coupled magnetic beads in the fight against COVID-19. Please visit our website at acrobiosystems.com for more information and to learn more about our more than 1,600 plus recombinant protein catalog, including immune checkpoint proteins and CAR-T targets. ACROBiosystems is ISO certified, and some of our COVID-19 diagnostic kits are also IVD CE certified. My email is prajwal.podel@acrobiosystems.com. For any other general inquiry or product information, you can contact us at order@acrobiosystems.com. Also, please visit our virtual booth and speak with our staff for more information about our products and services. Thank you, and I'll take any questions.